Hello, and welcome back to Introduction to Genetics and Evolution. In this set of videos, we'll be talking about evolutionary trees and the field of phylogenetics, the study of relationships among organisms. In this first video, we'll look at how you read evolutionary trees and how you understand the information within them. And then in subsequent videos, we'll look at how you generate evolutionary trees and some uses of having them. So let's go ahead and get started. Well, this tree of life analogy is one that many of you are probably familiar with, and we commonly conceive of the relationships among organisms in the context of this sort of tree of life. Now, this is a useful visualization for evolutionary history, and this can be done within a species among different individuals, almost like a family tree, or it can be done among closely related species, or it can be done among all species. And you see this dates back to Darwin's Origin of Species book. This was actually the only image in Darwin's book, and it shows this tree of life. Now, there's a couple of different meanings for trees. Now, here are two related meanings of trees. One is you can look at trees in the context of groupings of similarity or classification, or you can think of them in the context of ancestry relationships. Now, let's do these two separately first, and then we'll bring them together. So looking at the figure on the left here, we have a whole set of vehicles. Now, which of these vehicles are most similar to each other? Well, we have the two Honda cars. They're very similar to each other. They are motor vehicles, just like this Toyota truck over here, but they're clearly not the same category. You'd say they're a little bit less similar to the truck than they are to each other. And they're very dissimilar to these pedal vehicles way over here. So the tricycle and the bicycle, which are very similar to each other. So this would be a categorization just in terms of similarity, that these two are very similar, these two are very similar. This is kind of similar to the others, and they're all in this general group of vehicles. Well, that's just in the context of classification. Another way of looking at trees is in the context of ancestry relationships. Now, I've depicted this here with a piece of the royal family. We have Queen Elizabeth II here on the bottom, and we have several of her grandchildren. So we have Prince William and Prince Harry, who are brothers. We have Princess Eugenie and Beatrice, who are sisters, and we have Zara, who's their cousin. So if you look, these two are sisters. They're very similar to each other in appearance and in relationship. These two are brothers. They're very similar to each other in appearance and in relationship. And all of them are somewhat similar. They're more similar than somebody just at random from on the planet because they share this common ancestor of Queen Elizabeth. Now we can take this further to look at classification groups in the context of evolutionary relationships. Many of you are familiar with some of these basic classification groups. So let's focus on these animals here on the far right. Now these are all animals. There are no plants here, so they have one thing in common. They are all animals. Okay? The top five here, as you can see, the top five here all have a backbone. So they are all referred to as vertebrates. So I have that classified over here. They're all vertebrates. In contrast, the fruit fly there is an arthropod. The top four here are all mammals. They all give milk to their offspring, right? So they have something in common, and they're more in common with each other than with the rest. The top three are purely carnivorous. They only eat meat, so that's something they all have in common. And the top two, both are canines. They're both in the group that has dogs, for example. And then ultimately they are different, like the wolf and fox are different from each other. So you see we have these classification groups, and this hierarchical classification system that we use today was begun by Linnaeus way back in the 1700s. Now he didn't talk about this in the context of relationships, but instead he used it as a way of categorizing the diversity we see in nature. And you see it goes from the most general down to the most specific. Right? You see the different groups have different things in common. And we see this in this diagram that I made here, right? That carnivores have more in common with each other than they do with primates, like us. Mammals have more in common with each other than they do with birds. Vertebrates have more in common with each other than they do with arthropods, etc. So these classification groups actually imply evolutionary relationships, and this was one of Darwin's insights. That if we were to draw a tree of life, it may be along these lines, where the fox and wolf have a very recent common ancestor. Further back, you see the common ancestor with cats. Further still back, you see a common ancestor with humans. Further still, the herring gull, and further still, the vinegar fly or fruit fly. Now, this sounds fairly straightforward. In practice, however, you can get conflicting results depending on which characteristics you use for making this sort of classification system. So one of the examples I used before was vertebrates. So vertebrates are animals that have a backbone. If you were to categorize the animals here that you see, which ones have a backbone and which ones don't? Well, the guinea pig obviously has a backbone, and the wood duck has a backbone, whereas the ladybug and the spider do not. So this is very clear that these two are similar and these two are different. In contrast, what if you were to use a different character? What if you were to use wings? Well, here, the ladybug and the wood duck have wings, the guinea pig and the spider do not. That is clearly not the classification that you would imagine off the top of your head. So, you can get conflicting results. Well, how do you resolve these conflicting results when you use different characteristics for making these groups? Well, in part, you use the weight of evidence. That presumably you're not going to just categorize things just by having wings or not, or just by having a backbone or not. But there will be a number of characteristics, and you look at how many of them favor one set of relationships versus the other. And you also will look at what I'll refer to as new versus old characters. Now, I'm not going to talk about that in this video, but I just wanted to toss that in there because sometimes when I first start talking about these relationships, people get caught up in that and they say, but, what about this? So I just want to put that now, and we'll talk about how you generate trees later. So what are some of the assumptions in making phylogenetic trees? One is we're looking at the summation of relationships over time. I love this figure. This comes from a paper by David Baum, who's a friend of mine as well. So here we have 4 butterflies. You can add the parents there, and you see that there's some relationships among them. That this one has these 2 parents, this one has these 2 parents, etc. You can add earlier generations, so you get this sort of web of relationships. You can then look at the whole population. You see this web gets more scattered. You can look at the whole species, and ultimately you get to this evolutionary tree. So what we're looking at is the sum of relationships. Think of it as the family tree of every organism that has ever lived. It is possible to generate that family tree, but it's going to be very, very, very dense. There's going to be a lot of different relationships, and it's going to take a long, long time to go back to this evolutionary tree. And that's essentially what is reflected in the evolutionary tree. It's this summation of relationships over time. It is reflecting evolutionary history, or most importantly, just history. We are assuming common ancestry of all the species that are being studied for generating the phylogenetic tree. That is an assumption that we're putting into there. We're assuming that there are clean splits into 2 or more taxa. In other words, the tree bifurcates, it splits. It doesn't have to be just 2. It could actually split in 3 ways all at once. But we're assuming these sorts of splits over time. We're assuming the formation of new lineages. We're also assuming that once species do diverge, they don't actually come back together. Okay? So here's a sample evolutionary tree that I'd like you to look at, as well as some terms I'd like you to know. So first, just look at the tree itself. Now, this is a tree that you've seen before. So we have the wolf and the fox, which are canines, or sort of in the dog family, I guess you'd say. We have our cat, wolf, and fox, which are all carnivores. And we have our human cat, wolf, and fox, which are all mammals. So what are the branches of this tree? Well, the branches of this tree are basically these lines coming out from a point. So these are all branches, just like if you're looking at an actual plant tree. Right? So these are all branches along the evolutionary tree. Now nodes are intersecting points of branches. So right here we have an intersection between the branch going to cat and the branch going to the canines. Here we have a node that intersects the branch going to wolves and the branch going to foxes and the branch going to wolves. And of course we have a node back here. So what do these nodes represent? Well, let's think about this for a second. The nodes represent the branching point where you have a lineage that goes on to evolve into the modern fox and a branch that goes on to evolve into the modern wolf. So this is the point where foxes and wolves shared their most recent common ancestor. So back here, those entities that eventually would have offspring that go on to become foxes and wolves were the same. They had a single ancestor at the node and any time before that node. Same over here. At this point in time in the evolutionary tree, some individuals may have descendants that go on to be cats and some descendants that go on to be wolves or foxes. The root of the tree is the base. It's way down here and below that. So that is basically the point of a most recent common ancestor for every individual that you're looking at in the evolutionary tree. Now there are such things as unrooted trees, and I'm not going to go into those in this class, but you may see them in other contexts. So you've looked at this tree and you see that there's these nice diagonal lines, and you see the points where we have the nodes, these most recent common ancestors of particular subgroups. Now I want to emphasize that these diagonal lines are not necessary. There are other ways of representing evolutionary trees, and you see this is exactly the same representation as before, except now the nodes are represented by a horizontal line. Okay? It's exactly the same as before. We still have the most recent common ancestor of humans with cats, dogs, and wolves about 100 million years ago. We have a more recent one for cats with dogs and wolves, and a more recent still one for foxes and wolves. So again, there's different ways of representing it. Now we assess the relationships among individuals in this evolutionary tree by looking at who shares the most recent common ancestor. So even if you didn't know that foxes and wolves were canines, you could tell they're more closely related to each other than to any other individual on this tree, because when you look at it, they share a fairly recent common ancestor. In contrast, cats with foxes share a more distant common ancestor over here. So the most recent common ancestor of cat with fox would be right here, which is further back in evolutionary time than the most recent common ancestor of fox with wolf. Now I want to stress, because there's something that people mess up all the time, don't worry about who is next to who on top. Look at the most recent common ancestor. A common mistake is for people to think, for example, that, oh look, the fox is more closely related to the cat than the wolf is to the cat, because look, the fox is next to the cat. But no, where is the most recent common ancestor of fox and cat? The most recent common ancestor of fox and cat is here. Right? If you go back in time, where do you get the ancestor that's shared? Right there. What about for wolf and cat? Same thing. That's the more recent common ancestor. So I'm going to ask you a question. I'm going to get you to look at a couple of trees. I want you to tell me if these trees are exactly the same or if they are different in terms of evolutionary relationships. Obviously, they look different, but that's irrelevant. I mean, are they the same or are they different in the context of evolutionary relationships? You may want to pause the video because this is going to be an in-video quiz, so I suggest you hit pause and then think about it and then play again when you're ready. Well, I hope that wasn't too difficult. So when you look at this, what is the most recent common ancestor of fox and wolf? Well, it's right there in this tree. It's right there in this tree. What is the most recent common ancestor of cat with either of the two canines? Be right there and right there. And with humans, with all of them, be right here and right here. These are basically exactly the same tree. And if you look at the common ancestors, it's not like all of a sudden fox is more closely related to human or fox is more closely related to cat in one tree than in the other. That the point at which you have the most recent common ancestor is exactly the same. And that's important when comparing trees. Let me give you the example looking at, say, just a family tree. So looking at this family tree, you see exactly the same thing. That we have Prince Harry and Prince William off to the left. We have these two in the middle. We have this person off to the side. If you rotate this tree, this does not make William any more closely related to Beatrice than he was over here. It doesn't change the relationships. All you did was just rotate the depiction of the relationships. But when you look at where the common ancestors are, the common ancestors, or the nodes, are in exactly the same places here as they are here, and the common ancestor is here for all of them. So the relationships did not change. It's very important not to look at to see if the ends are near to each other. For example, these two are no more closely related here, or no more distant related here, than here. It doesn't matter who's next to each other. All it matters is where the most recent common ancestor is. And in that case, the most recent common ancestor for those two people is Queen Elizabeth in both cases. So let's look at this in a broader sense. Here's a whole set of phylogenetic trees, and all I've done in each of these cases is rotate the nodes. All of these are exactly identical. Every single one of these is exactly identical because, again, the most recent common ancestor for fox and wolf is the same. The most recent common ancestor for the cat with the others is the same. The most recent common ancestor for humans with the others is the same in each of these. These are all exactly the same tree. I'm emphasizing this a lot because I want you to see that it doesn't matter the ends. This is something a lot of students get confused in. But you see in each of these cases, the canines group together, right? The carnivores all grouped together. And then the human is always the most distantly related. So I'm going to show you now a set. here's a set of 4 phylogenetic trees. I want you to look at these. And you're going to pause the video again just a second because there's going to be an in-video quiz. I want you to look at these and tell me which of these is different. Well, I hope that wasn't too difficult. Let's work through it. So B and D are clearly the most closely related in all of these trees. Okay? Now, what's the next most closely related to B and D? Well, we look at the most recent common ancestor. Let's start with number 3 over here. There's the most recent common ancestor out from that, and that would group A with B and D. There, A with B and D. There, A with B and D. Here, whoops! This is C with B and D. This is a different phylogenetic tree from the other two. So, looking at which of these trees is different, A has a more recent common ancestor to B and D in trees 2, 3, and 4, but not in tree number 1. So, tree number 1 is the one that's different. Let me give you another try. I'll warn you ahead of time, you may consider it somewhat of a trick question, but I hope it helps you understand the concept. Looking at this, which species is or are most closely related to species A? So pause the video, think about it for a second, and then take the quiz. So this was a little bit tricky. Now, here's A, and I'm asking which species is most closely related to A. So let's go to the common ancestor. There's the common ancestor of A. There's a node between the branch leading to A and the next branch out. And what you see is this branch goes both to B and to D. There is no difference in average relationship between A and D than between A and B because they share the same most recent common ancestor as depicted right there. So I hope that was useful for you. Now, let me just talk very briefly about a couple of applications of evolutionary trees. We'll come back to another one in the third video in this series. Now, generally speaking, evolutionary trees are of course used to classify nature and understand the process of evolution. You may want to know, for example, is a fox more closely related to a wolf or to a dog? Well, by making an evolutionary tree, you can find out which of those is more closely related. Now, there are more specific applications both in medicine and in law. Now, medicine, many of you are familiar with the process for identifying flu strains for the vaccine. That does involve phylogenetic reconstruction. So this is a direct application of evolution to health, that you want to see what is the diversifying strain of flu and make sure that gets into the vaccine. In terms of law, there's some interesting cases out there where people have traced various infections, such as with HIV, to a source. And here are 2 court cases mentioned in this context. And in cases where people knowingly infected others, you need to establish that others contracted the virus from the accused and not from elsewhere. And in doing that, evolutionary trees can be used to show that the victim strain of a fast-evolving virus is much more similar to the accused strain than to any of the others in the population. And that can essentially prove, at least with statistical confidence, that the victim got the disease from the accused and not from somebody else. And then if you have data showing that the accused knew that he or she was infected prior to that, then clearly they are guilty of intentionally withholding that information and passing on the disease. So that's one application of phylogenetic trees to law, and again we talked about one for medicine. Well, I hope you enjoyed this piece. In the next video we'll come back to how you generate these evolutionary trees. Very exciting. Thank you so much. Hello, and welcome back to Introduction to Genetics and Evolution. In this set of videos, we're looking at evolutionary trees and the study of phylogenetics, or the relationships among organisms. In the preceding video, we talked about how to read an evolutionary tree. In this video, we're going to look at generating evolutionary trees. And I'll give you the opportunity to generate your own evolutionary tree using some DNA sequence data by the end of this video. So let's get started. What do we need to generate an evolutionary tree? Well, we need to look at a set of traits and decide on similarity among the organisms being studied. So this is a set of 4 organisms that I showed you in the preceding video. So what traits should we use? Well, I picked here just a random set of 4 traits, and you'll see that the answer isn't totally straightforward of what we do. That we can look at the exterior and say hairy versus having feathers versus smooth. Well, how would this categorize these? Well, this one's hairy, this one's got feathers, this one's smooth, this one's hairy. So we should say the spider and the guinea pig are more closely related? That's a little bit funny, huh? Well, let's look at the others. How about which ones have a backbone versus not? Well, having a backbone would be these two have a backbone, these two do not. That might be okay. What about something else? What about number of legs? Well, here we have 8, here we have 6, here we have 2, here we have 4. Well, that's not useful at all. And the last thing is, what about presence or absence of wings? Well, that was the example I showed you in the preceding video, that these two have wings and those two do not. So you can see it's not clear what to do when you're looking at a single character, and you can in fact be misled when looking at single characters. So the way this is addressed in practice is that we try to study the weight of evidence, especially by looking at multiple characters. Right? That we're looking at the number of characteristics that favor one set of potential evolutionary relationships versus another. And I'm going to talk about this first. Then we're going to come back to looking at new versus old characters, and I'll just make a quick comment at the end about some more in-depth models. So, with respect to similarity, here is the same set of animals that we looked at before. And here I've laid out a bunch of different characteristics, and these are ones that are potentially useful. And what do we see when we look at this? Well, taxon 2, which is the ladybug here, and taxon 3 being the wood duck, they share the fact that they both have wings. Okay? In contrast, the spider and guinea pig do not. For all the other characters, you'll notice that taxon 3 and 4 are similar. These both have an endoskeleton, their skeletons inside their body, whereas these both have an exoskeleton. These two are warm-blooded, whereas these are cold-blooded. Though that's a little bit of a cheat that I'm not going to talk about in this video. These two have closed circulatory systems. These two have open. These two have simple eyes, whereas these two have complex eyes. And these two have a backbone, whereas these do not. So, on balance, when you look at the weight of all the evidence here, you see there's a strong relationship between taxon 3 and 4 as being similar, and 1 and 2 being different from them, even though this category of wings throws that off. But I have to emphasize that similarity is not enough. You can't just make a good evolutionary tree based on similarity alone. Instead, we end up having to look at new versus old characters. Characters that evolved fairly recently versus ones that may have been retained since the most recent common ancestor. Now let me show you why this is true. Similarity alone can be misleading when you're looking at old or putatively ancestral characters, characters that have been around potentially since the root of the tree. Now let's imagine your trait is body color, and let's say the two body colors that we have here are black and red. And let's say the ancestor was black, and let's say the color of the branch that I put on here is the phenotype or the appearance that was present at the time. And we have a new mutation to red that arose right here in the lineage leading to C and D. Okay? If you were to go by similarity alone, C and D are very closely related to each other because they're both red. And then A, B, E, and F are all closely related. Well, is that true? Look at the tree for a second. Is that true? The answer is partially. It is true that C and D are closely related, as you can see over here, but A, B, E, and F are not particularly closely related. You see that, in fact, B is more similar to C than B is to A, right? Because the most recent common ancestor of B and C is right here, whereas B and A is further back. So this works for new or derived traits that have arisen during the evolutionary time of the tree that you're looking at. This now assumes that changes are infrequent. And again, this tends to work just for the newly evolved or derived trait. Now, how can we leverage this to actually study evolutionary relationships? The fact this only works for the derived traits and identifying which are most closely related. So this leads to what's referred to as the parsimony method for tree building. Basically, what we're going to do is we're going to compare the traits to what we believe the ancestor would have had in terms of the trait. Now, typically speaking, the ancestor is not usually still around. We don't usually get to look at them. So what people do in practice is they look at what's referred to as an outgroup. So, for example, let's say you were looking at a phylogeny of insects. Let's say these were all different insects. The outgroup you might use might be, for example, a spider. So something that's similar but clearly not an insect, so it's just outside there. Using the spider and then looking at all these insects, you would then try to identify which forms for each trait are ancestral, or basically have been around since that common ancestor, and might, for example, be present in spiders as well, or if they're derived, a trait that has arisen within the evolutionary history of insects that you're trying to study. So what you're going to do then is you're going to construct a tree using the minimum number of changes from the ancestral form to the derived form. Right? The minimum number of possible changes. You'll be using, for actually looking at relationships, you'll be using what we refer to as shared derived traits. The technical term for this is synapomorphies, but don't worry about that. You're looking for shared derived traits. So in this tree right here, the ancestral trait is black, right? Black body color. The derived trait is red. So which individuals share the derived trait? The derived trait is red. The individuals that share it are taxon C and taxon D. So the assumption here is that the simplest tree is correct in terms of these derived characters and looking specifically at those. So that places C and D as being close relatives. This tree also uses the minimum number of changes because it only requires one change. So what would have happened instead? Let's say, so we have A, B, C, D, E, F, and you see that here's the common ancestor. What would happen if instead C was more closely related to F, for example? Well, let's take a look at what that trait would have to look like. Well, there it is. Now C is most closely related to F, D is more closely related to E. I just changed the tree around. This, in contrast to the preceding one, requires two changes, right? We have to have a mutation from the ancestral form of black to red in the lineage leading to C, and a mutation from black to red in the lineage leading to D. This requires two mutations now. Why is that? What if the mutation was way back over here somewhere? Well, if the mutation happened way back here, E and F would also be red. But instead, we just have D and C being red. So this now requires two mutations. So this is clearly not the most parsimonious tree. Now, I won't say this tree is not possible. We don't necessarily know the simplest answer is always correct, but it's not the simplest possible tree. Now, an important thing to keep in mind here is all this is showing the fact that C and D have the shared derived character is that they are more closely related. It actually says nothing about the rest of the evolutionary tree. So what about A, B, E, and F? Well, we can move them someplace else. Now I put E and F over here and A and B over here. This is actually just as parsimonious as the first tree I showed you. So all you are able to infer from looking at all this is C and D are close relatives, and you don't know the relative relationships of A, B, E, and F. So ultimately, making a tree using the parsimony method requires that you have to use multiple characters. Or if you're using, say, DNA sequence, you use multiple nucleotide positions within a DNA sequence to place all the species you're studying onto the tree. Okay? Because you can only look at those shared derived characters. So let's do an example. So this is a pretty straightforward example where we have an outgroup species, right? And we have its set of characters for a particular trait. Now we're going to use the outgroup species to infer which form of each trait is ancestral or derived. Okay? We have 4 species. Let's go ahead and draw a tree based on what we get here. So we already know the outgroup is the more distantly related. Right? That's just defined as such. So when we look at these different traits, what do we see in terms of these shared derived characters? That's the most important thing we're looking for, shared derived characters. For trait number 4, the derived character is only found in species A. So it's not shared. It's not a shared derived character. It's just a derived character found in species A. That's actually not useful for phylogenetic inference at all. Just draw a line through that. We'll actually come back to that in just a second, but it's not useful for phylogenetic inference. Trait number 3, what do we see? The derived character is 2 legs because the ancestral form might be 4. And what we see is that all 4 species share that. That's actually not particularly useful either for phylogenetic inference. So we might say right here there's a transition in trait 3 from having 4 legs to 2 legs. We don't actually know it's right there. It could be in the lineage going to the outgroup too. But just for ease of argument, let's just put it right there. So that doesn't really help us a whole lot. What about trait 2? Well, here, the outgroup has 4 arms. Species D also has 4 arms, but these 3 have 2 arms. So we have a derived form, 2 arms, and we have it shared by species A, B, and C. So given it's shared by A, B, and C, that would infer that D is more distantly related because it does not share the derived character. Okay? So we have a transition point right here for trait 2, going from 4 arms to 2 arms in taxa A, B, and C. For the last one here, our outgroup has 4 eyes, species D has 4 eyes, and species B has 4 eyes, but A and C share the derived character. Okay, so in that case we can say that B comes out over here, and A and C are the closest relatives. So we can place in here that extra transition for trait 1 right there. So this shows all the transitions. So here's the point in time right there on the evolutionary tree where trait 1 transitions from 4 eyes to 2 eyes. Here's where trait 2 transitioned from 4 arms to 2 arms. Here's where trait 3 transitioned from 4 legs to 2 legs, possibly, or could be on this other branch. And then I mentioned trait 4 over here. We can actually go ahead and place it on there. That would be right there. It doesn't particularly help evolutionary inference, but we can place it on the tree nonetheless. So, what would happen if we add a character that doesn't agree with the other characters? Well, here's trait 5. When we look at this, we have the outgroup having no wings, and A and D share the derived form. A and D were not sister species, as you saw before. So let's look at the possible alternative trees here, and let's place all the characters on there and see how many transitions it takes. So the left version of this has the derived version of trait 5 arising only a single time. So here it is, trait 5 arises once, and it clusters in A and B. Or I'm sorry, A and D, I said that wrong. So what does this do to the other things? Well, because of this, if we assume that this one is right and the others are wrong, then traits 1 and 2 have to have arisen twice somewhere in the tree. And it can't have just arisen once, because if we assume trait 5 is correctly reflecting evolutionary history, then traits 1 and 2 have to have arisen twice. In contrast, the tree on the right assumes that first tree that we saw, and this is assuming that trait 5 arose twice. So here's trait 5 once, here's trait 5 a second time, and that's what makes A and D similar to each other in that context. But here we have traits 1, 2, 3, and 4 all arising only a single time. So how many changes do each of these require? Well, let me clear all these circles so you can see it a little bit better. Well, over here we have 1, 2, 3, 4, 5, 6, 7 changes, right, to make this evolutionary tree. In contrast, this one over here requires only 6 changes. So which of these is more parsimonious? The answer is the one on the right is slightly more parsimonious because it only involves 6 changes rather than 7 changes. Now, the way I did this, there's other ways you could actually make the same tree if you have changes going, for example, to one form and then back. But no matter how you do it, this tree is slightly more parsimonious because there's no way to make the tree such that it would involve fewer than 6 changes. Okay? So let me give one for you to try. Here's one for you to try. This now I've changed a little bit, so now this involves DNA sequences instead of wings or no wings, 4 legs or 2 legs, but the principle is exactly the same. Now you may be slightly panicked. What? How do I do this? So let me break it down for you. First thing you do is you just look at what are the sites that are variable, right? Well, these are the sites that are variable on the tree or on the sequence. So we have traits. This is equivalent to saying trait 1, 2, 3, 4, and 5. Okay, so that's pretty straightforward. And what you're doing is you're comparing the outgroup to the 4 species, and you're seeing which of them share the derived form. And then from that, I want you to make an evolutionary tree. So why don't you pause the video for just a second, try to construct the tree, and then I'll give you 3 choices, and then I want you to do the in-video quiz to try each of the 3 choices, or just to see which choice it is. Okay? So go ahead and stop the video for a second and construct the tree. Hopefully you've already stopped the video. I'll go ahead and show you the 3 choices now, and then you have to vote which of the choices it is in the in-video quiz. Here are the 3 possible choices. Well, I hope that wasn't too difficult. So again, what are we trying to do? We're looking at the shared derived forms. So I've highlighted here the derived forms for each of these things. And as you can see, trait 2 is not useful because the outgroup has C, everybody else has C except for species B. So this is actually, this does not have a shared derived form, so we can just ignore this one. For the others, trait 5 is also not particularly useful because all it has is the outgroup differs from the 3 non-outgroup species, so we don't really need this either. The ones that are more useful are traits 1, 3, and 4. So who shares the derived forms? Well, for trait 1, A and D share the derived form, so we can draw something that clusters A and D, right? For trait 4, again, A and D share the derived form. So again, A and D. So if we were to mark on the tree, this is where trait 1 changes, and this is also where trait 4 changes. And then, what happens with trait 3? Well, that clusters B and C together. Right? So this would be the evolutionary tree that we'd expect. That B and C are clustered together by trait 3, A and D are clustered together by traits 1 and 4. And theoretically, if we were to put the other things on there, this is where trait 5 would have transitioned, or potentially in the outgroup lineage. And right before species B, you'd have the trait 2 one, if you were to place it all on there. But essentially, B and C are sisters, A and D are sisters. The question to ask is the answer right? Well, the real answer to that is we don't know. Parsimony doesn't always result in a tree that accurately reflects what happened over evolutionary history. Because you can have similarity in some traits that doesn't reflect ancestry, but reflects a convergence, that you had 2 mutations that happened to go to A at the same site that made them become more similar. That's often referred to as convergence, and those traits are often referred to as homoplastic, right? You basically can't have these shared derived traits evolving more than once, and it's something that there's no way around. Now, the way people have tried to address this is to use various model-based methods, such as maximum likelihood methods or Bayesian methods, and those often do better, but they're still not always correct. They're not 100% correct. And different genes may give you different trees. So in the next video, we'll talk about an application of phylogenetic trees for testing evolutionary hypotheses, under the assumption that your tree is correct, or at least very likely to be correct. Thank you. I hope you enjoyed this video. Hello, and welcome back to Introduction to Genetics and Evolution. In this set of videos, we've been talking about evolutionary trees, also referred to as phylogenetic trees. In the first video, we talked about reading trees and interpreting them. In the second video, we talked about constructing them. In this video, what I'd like to do is I'd like to talk about one use for evolutionary trees, and specifically using the phylogenetic comparative method. Now, in the first video, you might remember I did talk about a couple of uses for trees, and certainly there's a general usefulness for them for trying to categorize life and understand the history of life on the planet. But I want to talk briefly about using this information for testing evolutionary hypotheses. Okay? Well, there's a lot of evolutionary hypotheses out there that can be tested, and I just wanted to present to you a couple that have been out there for a long time. These are 3 broad patterns that have been described since the 1800s. They've mostly been applied to warm-blooded animals. They all have specific names, but I don't want to burden you with too many names. One is that, typically speaking, in colder climates, animals tend to have a larger body size. Okay? Another is that in colder climates, animals tend to have shorter appendages. By appendages, I mean things like arms, legs, but even things like ears. So this is an example right here. Notice how small the ears are on the polar bear relative to the grizzly bear. So they seem to fit that second rule. And last but not least, animals tend to have, in general, a lighter color in drier climates. Now, these are tendencies that are out there, and people have seen them, but how do you test whether these rules are general and whether they actually have some sort of underlying cause, whether they're meaningful in a sense? Well, the most basic thing people would do is look for multiple examples, right? That if you tend to see a lot of animals have short appendages in cold climates, you'd say, oh, okay, this fits the pattern. And you see many animals have long appendages in warm climates, again, fits the pattern. So you'd have a seemingly general pattern, and you would have some potential underlying cause that you're interpreting in this case to be climate and temperature in the climate. But there's a problem. And that problem is the problem of independence. So let me give you a simple example. Let's say you have 2 groups of people. One group that exercises, and I have them here in blue, and one group that does not exercise, and I have them here in red. So that would be equivalent to temperature, for example, in the previous thing. And you sample a lot of individuals, and look at that. There's all the blue individuals, they tend to be kind of tall. There's the red individuals, they're not quite as tall. So there appears to be a pattern there, right? Well, the issue is, are those 2 things independent? But what if all your exercisers are relatives? Well, then you don't really know, are all these blue exercising individuals tall because they're exercising, or is it that they just happen to have a tall allele at a particular gene, and then these individuals happen to have the short allele at a particular gene or a set of genes? That basically we can't separate out this relationship, this historical familial relationship, from the fact that whether they exercise or don't exercise. Well, this illustrates the problem of independence. Now what happens here is you have a pattern, but this pattern may be an artificial pattern because the groups are relatives. And basically the differences you're observing between these groups may have nothing to do with the treatment. The treatment in this case could be exercise, it could be cold or dry climate, but you can't really tell if all these individuals here are short because they experienced lack of exercise or a particular kind of climate, and these individuals are tall because you have this confounding variable. You have this extra facet in that these are all relatives, these are all relatives here. Okay? This is sometimes referred to as pseudo-replication, when you have sampling multiple individuals that differ in a particular treatment, but in fact they are not independent of each other. That what could have happened theoretically is that there is a genetic predisposition to being short and a genetic predisposition to being tall, and let's say for example there was a transition here to becoming short. There could be just one evolutionary transition. In this case, the real sample size is 1. Your number of what are referred to as phylogenetically independent contrasts is 1. Because there's only one transition between blue and red when you look at this branching thing. Now, we don't know whether it's a transition from blue to red or from red to blue, but all you need is one transition. Now, in contrast, notice here we have 4 short red individuals, 2 tall blue individuals. In this case, we have exactly the same number of individuals, but we don't have this confounding aspect of relationships because we have a single family that has both blue and red. We have a single family here that has blue and red. We have a single family here that has blue and red. What is the minimum number of transitions it would take to get this? Well, it would take at a minimum 3. Let's say, hypothetically, let's say the ancestor was red. Then you have a transition to blue in this family, transition to blue in this family, transition to blue in this family. Okay? So in this case, the real sample size, or the number of phylogenetically independent contrasts here, is 3, because that's the minimum number of evolutionary transitions that you had in this example. And if in this case, all these tall individuals exercise and all the short individuals don't, then you have slightly more confidence in that exercise is leading to a higher height. So let me give you one for you guys to consider. Imagine a hypothetical scenario that you want to test that perhaps slugs retain more fat in high latitudes, potentially because they're colder, than in low latitudes, and maybe they do this for insulation. So what you've done is you've sampled 6 island slug species, 4 of which are from high latitudes, 2 from low latitudes. Okay, you've sampled them from you know their latitude and you know their fat content, and then you've gotten DNA sequences and you constructed an evolutionary tree using some of the methods from the preceding video. And let's say this is the tree that you got. So this the blue indicates the latitude, so these 4 are from high latitude, these are from low. The red letters indicate the fat content, so those 4 have high fat content, those 2 have low fat content. And you have this evolutionary tree, this phylogenetic tree. How many independent contrasts do you have? So think about that for just a few minutes, and then you can pause the video if necessary, and then answer the in-video quiz. Well, I hope that wasn't too difficult. Looking at this, again, when you're trying to think of how many phylogenetically independent contrasts, you try to think what is the minimum number of changes in state. So let's say, hypothetically, way back in time, the ancestor was at high latitude and high fat content. Well, it only takes one transition for both those traits right there in order to get this full set of relationships. So in fact, for this tree, you only have one phylogenetically independent contrast that you know of. Now, this is a very important thing, and evolutionary biologists were a little bit slow to fully accept this until maybe the 1980s, where it was really brought to people's attention by this fellow here, Joe Felsenstein. And he had a great quote in one of his papers: "Phylogenies are fundamental to comparative biology. There is no doing it without taking them into account." Now again, when we're trying to make broad evolutionary inference, like those 3 rules from the beginning of the video that I presented to you, we need comparisons among groups. But in order to assess our confidence in these comparisons, we need to know evolutionary history. We need to know how many independent comparisons we're looking at. So comparisons are most meaningful when studied in the context of their evolutionary history. and that's where the field of phylogenetics comes into play. It is very, very important for evolutionary studies. Well, I hope this set of videos was helpful, and I hope you had a good time. Thanks so much.